Yeah, so far, so we talked about, so we looked at why, proteins are affected, protein structures and dynamics are affected by pH. And let's take a look at the protein structures a little further. So as you mentioned, so the amino acids are connected through peptide bonds, but as you know from the chemical, your chemical knowledge, actually this peptide bond forms, this carbon and peptide bonds is a sp2, has a sp2 hybridization, and this nitrogen also has sp2 hybridization. So these peptide bonds are forming a conjugated bond, and they are on a plane. They are forming a planar structure, right? So they are forming a planar structure. So they don't rotate. So this structure, so these four structures don't rotate. They are just on the same plane, right? But instead, so this bond and this bond and this bond can be rotated, right? So these are rotatable bonds. So as you see on this carbon and this carbon is a sp3 carbon. So these bonds are rotatable. So the bond between this N and C, and this C is called, we call it as a C alpha, because so there is an R group. So the R group starts with a C beta, right? Oh, I wrote it wrong, right? So this C is called C alpha, and this R group starts from C beta. And as I mentioned, and this bond and this bond are rotatable, and this bond is called, and this, so that this angle, so that this bond, so the dihedral angle that forms this angle is called defined between, so the dihedral angle formed between, this atom, these four atoms are called phi angle, and also another dihedral angle formed between this N, C alpha, C, and N is called psi angle, okay? And these two are rotatable. So based on these rotations, and so the protein can have, because these two angles are rotatable, the whole protein chain is flexible, and they can adapt various structures. And in addition to these phi psi angles, and some amino acids have rotatable angle too. So the rotatable angles in side chains are called chi angles. So the first one is a chi 1 and the second one is a chi 2. So as you have learned from the organic chemistry class, and so actually the dihedral angle is defined between this carbon atom and the carbon atom behind, right? And based on the rotation of this dihedral angle, so the energy can be changed. So the energy maxima and the energy minima. These staggered conformations have lower energy than these eclipsed forms, right? So based on the rotation of these dihedral angles, actually the energy state of the whole protein chain can be changed, right? And so to represent, these phi psi angles, there is a plot called Ramachandran plot. So that this Ramachandran is adapted, so this Ramachandran plot is named after a famous protein structure scientist, Ramachandran, who first suggested this plot. So in this plot, so the x-axis is about the phi angle, and the y-axis is about the psi angle. So they are plotting, which phi psi angles are possible. So he investigated the protein structure and plotted this plot. So here the darker colors means that, I mean, these are popular. These are, highly populated region. So which means that, so these combinations of these phi psi angles are preferred, and pale colored means are less preferred. And these white regions are forbidden. So these white regions are forbidden region. Okay. So then why are these kind of difference? As we mentioned before, so the dihedral angle actually determines its energy, right? So these eclipse forms have higher energy, so the higher energy conformations prefer to have lower energy, right? So based on mostly because of steric hindrance between atoms, so that there is a certain preference of phi psi angles, and people realize that this region corresponds to the alpha helical region, and this region corresponds to beta sheet region. So that's why there are two popular, local structures, secondary structures, alpha helix and beta sheets, and other regions are not preferred. Okay. Then there is once Anfinsen found his principle, performed his experiment, then another scientist called Levinthal actually suggested a paradox. So if a chain of 100 amino acids is considered, and it is assumed that each amino acid can have one of three conformations, like extended, helical, or root. So the extended is the synonym of beta sheet structure. So the helical structure means the alpha helix. And loop structure it's neither beta sheet nor a alpha helix. So it's not beta sheet and it's not alpha helix. So usually, the local structure, so the local structure of a protein can be classified into three classes largely. Then there are, 3 to the 100 possible ways to arrange this chain, right? So if one amino acid can have three structures, then if we connect, 100 amino acids in side by side, then the whole chain can have 3 to the 100 possible structures. And this is roughly 10 to the 48 conformations, and bond rotation of an amino acid, or the chemical bond, can be estimated to occur at the rate of roughly 10 to the 14 inverse second. This means that the search for the right conformation through random searching alone would take the order of 10 to the 34th second, which corresponds to 10 to the 26 years, which is several orders of magnitudes greater than the age of universe. Okay, so which means that so if protein is folded at a random fashion, if protein is just randomly folded, then it takes super long time. But actually proteins are actually folded generally within milliseconds or within microseconds. Some proteins are folded within microseconds. So this is a paradox. Then it means that proteins know how they should be folded so that they can be folded very quickly, right? So this is the problem of protein folding. So the Anfinsen suggested that the proteins must fold in a highly efficient way, right? So they should be folded very quickly, generally, folds within microseconds or milliseconds. So from unfolded structure to some specific structure, okay? So one, at-hand example is that think about folding a snake puzzle like this. So you must know a snake puzzle you must have seen, wooden puzzle like this, right? Then if you think about, folding this to this final structure, then if you don't know how to fold this, then you should try, many different times, right? So then it would take very long time. But if you have to fold this from here to here, within, milliseconds, then in order to do that, so you definitely should know, answer. If you know the answer, if you know the exact order, then you can fold it, very quickly, right? But if you don't know the answer, then it'll take very long time to search, the right path, the right, folding path, folding way. So identifying how and why proteins fold in a specific mechanism is called protein folding problem. So how and why proteins are folded in such a way, so the revealing that, investigating that question is called the protein folding problem. And this protein folding problem is one of the most challenging problems in biophysics and biochemistry. So still, so many people, many researchers all over the world are trying to understand the protein folding process. So why proteins are folded in such a, predetermined structure, and how they can be folded so fast. Then why do proteins fold, in terms of energetics? As we have learned from the thermodynamics, and all chemical reactions occur to a direction where Gibbs free energy decreases, right? So that all the spontaneous chemical reactions happens to the direction where Gibbs free energy becomes lower, right? Then if we think about a protein, if we think about a chain molecule, then when protein is unfolded and they are freely, rotatable, then delta S is large, right? So the entropy is large. But when protein is folded, then protein is, folded to a specific structure, and they don't, rotate as much as when they are unfolded. So which means that their enthalpy is large. But instead, so the atoms are, closely contacting to each other. So which means that they have, lower electrostatic energy, and also they have lower van der Waals energy, right? So it means that their enthalpy is large. Delta H is large. So this explains why a protein is unfolded when temperature is high. So if you think about the, delta G, the equation of delta G. So delta G is delta H minus T delta S, right? And spontaneous reactions occurs to a direction where delta G becomes negative, right? So when protein is folded, then delta H, the contribution of delta H is large, but delta S becomes low. But when protein is unfolded, then delta H becomes low, but delta S becomes large, right? So if temperature becomes higher, then the contribution of this entropy term becomes larger, right? So the reaction goes to where delta S becomes larger, right? Because delta G has a negative sign with the entropy term, right? So that when T is high, so the contribution of entropy becomes more important, right? So protein likes to be unfolded. But on the other hand, so when T is low, then the contribution of enthalpy becomes larger. So the enthalpy becomes more important. So the protein likes to be folded, because when they are folded, their interactions between side chains become stronger, right? So if you think about what you have learned from the, general chemistry class and thermodynamics class, and so you see that, there is this kind of the relationship, right? So and in terms of chemical reactions and also in the perspective of Gibbs free energy, so the native structure of a protein corresponds to the lowest Gibbs free energy structure among all possible structures. So if you draw it like this, then the native structure has very low energy, but also has a very low entropy. But on the other hand, unfolded structures, so this here corresponds to unfolded structure, right? Unfolded structure has high energy, also high entropy, right? So the protein folding event actually corresponds to, among this table so the protein folding event actually corresponds to the fourth column, right? So when protein is folded, delta H becomes negative, energy becomes low, and also at the same time, entropy becomes low. So the protein folding generally happens at low temperature, and unfolding generally happens at high temperature, right? So this explains the the overall thermodynamics of protein folding process. Then what determines the energy of a protein structure? And there are several terms, but if we ignore quantum mechanical contribution, then largely so the energetics which determines protein folding can be classified into 1, 2, 3, 4, 5, 6 terms. And among them, probably the most important term is about the hydrophobic effect. And the next important factors are like hydrogen bonds and ionic interactions and disulfide bonds, van der Waals, and dihedral angle energies. So let's talk about hydrophobic effect first. So the hydrophobic effect is that the polar groups likes polar solvents, right? As you have, so the one of the old wisdom of chemistry is that the likes dissolve likes, right? So which means that polar solutes dissolve in polar solvent, and non-polar solutes dissolves well in non-polar solvent, right? But usually protein is solvated in water, right? Your body mainly consists of water, right? So the proteins generally, but as we mentioned before, so some amino acids are non-polar and some amino acids are polar. So which means that the polar side chains likes to be exposed to the solvent, but instead non-polar side chains likes to be buried inside the protein, right? So that's the one of the main driving force of protein folding. So the exposed polar groups likes to interact, these polar groups likes to interact with water molecules, right? So this is the one of the main driving force of protein folding, and the polar amino acids likes to be exposed, so polar side chains, and non-polar amino acids likes to be buried. And also there are non-bonded interactions. Non-bonded means them non-covalently bonded interactions. So the first one is the hydrogen bond, right? So the hydrogen bond is between hydrogen connected with with N or O, and the interaction with this O, like these interactions, right? These interactions, so the hydrogen bond interactions are very strong. So the strength of hydrogen bond is actually between it's weaker than covalent bond, but it's stronger than just the electrostatic energy. So the hydrogen bond is very strong. I mean, it's nearly covalent bond interaction. And the salt bridges are also called, I mean, another contribution is the salt bridges. Salt bridges is also called as the ionic interaction. So in other words, I mean, it's an electrostatic interaction. Right, electrostatic interaction is between charge Q1 and Q2, and it is defined like this, right? So if two particles have charges and they are interacting strongly. And another interaction is a van der Waals interaction. I mean, as you know, I mean, so every atoms have van der Waals interactions, just whether it is polar or non-polar. And van der Waals interactions just act between all atoms, and as you know, I mean, so the van der Waals interaction has a form of like this. I mean, so if you draw the van der Waals interaction, they look like this, right? So the x-axis is a distance between atoms, right? So which means that, I mean, just the van der Waals interactions, they just like to be close. So the atoms just like to be close. I mean, so if atoms are just at a right, point, I mean, they just want to be stick together. So this van der Waals interaction happens mostly between, bulky side chains, right? Because, I mean, bulky side chains tend to have more hydrogen, have more van der Waals interactions because they have more interaction sites. And also there is another, factor that affects protein structure and its folding is called disulfide bond. As you know, the disulfide bond is a bond between sulfates. And S and S can form, a covalent bond. And there is only one amino acid which has sulfur in it. So it's a cysteine. And if you remember, then the cysteine has SH group at its end, and if two SH groups come closer, then these hydrogens are, extracted, and the covalent bond between these sulfur groups are formed. So there could be multiple, disulfide bonds in one protein, and as you see, I mean, so these cysteines can form, disulfide bonds between amino acids. And as I mentioned, I mean, so these disulfide bonds are covalent bonds, right? So these are covalent bonds. So they are quite strong, and so they are, so once these, disulfide bonds are formed, I mean, they rarely become broken, and so they just keep this distance, right? So they play an important role in stabilizing a protein structure. And lastly, so there is an interesting protein called, protein chaperone. So as I mentioned, so although, most proteins, most protein folding events occur correctly, but some proteins, so during, protein synthesis, some proteins are generally, incorrectly folded, or they, remain unfolded. In such cases, some proteins need help to become folded. But some proteins need help to be folded. So in that case, so protein chaperone, helps. So once, unfolded protein happens, then this unfolded protein goes inside the another protein called protein chaperone, and this protein chaperone helps a protein to be folded. Then once it is folded, then protein chaperone, expels, this, put out, this, protein this folded structure. Then this one becomes, active, and this one works inside the cell and inside the living organism. So the protein chaperoning proteins are proteins which help other proteins get folded to its native structures. So the one important example is called GROW-EL and GROW-ES system, and there are many other protein chaperoning systems in living organisms, so which is also a very interesting topic of research. So far, so in this class, so we talked about the basic of protein structures, and we talked about what is the protein folding problem. And this protein folding problem is a very interesting problem and still remains a very active, topic of research. So in the next class, so we're going to talk about how to use, some protein structure modeling softwares and web servers. Okay, so let's call it a day.